So we're going to begin with the synthesis of macromolecules. And there is a whole word for that. So when we say synthesis, we are making, we are synthesizing, we are generating, we are compacting, storing, building up. So that is known as anabolism. So metabolism refers to all the biological activities that happen in the cell, that is energy wise and energy related. So catabolism is when I break down macromolecules into micromolecules. That is catabolism. While anabolism is when I build up micromolecules into macromolecules. So if I bring little bricks, and I make a wall with those little bricks, am I building up or am I breaking down? Building up. And what word do we use for building up? That is anabolism. And if I have a wall that I'm going to disintegrate and break into little pieces, what am I doing? That is known as catabolism. Okay, the same thing. When I eat my sugars, the sugars come in the form of polysaccharides. They are so big. Disaccharides. So I break them into monosaccharides in order to get energy out of them. But if I already have enough energy, I'm no longer burning them, then I store them. So in the fridge, you store the food. When you need it, you break it down. When you are full, you store it back in the fridge. So the same thing, your body is like that. It performs anabolism to store all the micromolecules in a big storage molecule, and we also have the catabolism, and that we break it down. So for example, this is a dynamic process. If you are sitting down on the couch compared to somebody who's running a marathon, that person who's running a marathon need energy, so they will be breaking down, catabolizing the macromolecules. And if you are just relaxed and you're eating, but you're not burning what you're eating, then you're going to be storing those macromolecules. And that's going to be known as anabolism. This is really important. So you need to know yes.
So molecules in general, whether it's macro, micro, or whatever, it's just molecules, the buildup of molecules.
No, you have to understand that micro is little, macro is big. So we use micro, we stack them up through anabolism to make macro. And we break down macro through catabolism in order to make micro. So now you have to understand that water has very important properties. What are the properties of water we discussed last week? The water has the ability to solvate. It's a universal, versatile solvent. Water can dissolve everything. Most of the solutions are dissolved by water. Obviously, they have to be polar solvent. And then there is non-polar subjects. These are not dissolved by water. These are dissolved by detergents. This is dissolved by lipids. So we now gonna look at how the macro and micromolecules do. How do they? How do I hide that? Everybody knows how to hide that. Okay. So the food provides nutrients like biological macromolecules that are necessary for life. But you have to understand we have four different types of macromolecules. What are the four different types of macromolecules that we have? We have excellent. We have carbohydrates, carbohydrates, and these carbohydrates are known as polysaccharides, and the polysaccharides are made up of disaccharides, and disaccharides are made up of monosaccharides. Disaccharide. Did you use a different color? Do we have? Okay. Okay. So monosaccharides come together to give me disaccharides, and disaccharides come together to give me polysaccharides. So going up, building up, that is known as anabolism. Breaking it down, that is known as a catabolism when we break down. So when do we get energy? If we are building up, we get energy, or when we are breaking down, do we get energy? When we are breaking down, you eat the food, you break it down, then you get energy. Then when you have enough energy, but there's still some food in your body, you build it up, you store it in the muscle, the sugar, and you store it in the sugar in the liver, and you store the fat in the abdomen. You will get a video summary to summarize that. You're gonna get a video summary to summarize this particular very important concept. Okay, so that is one, carbohydrates. Don't forget that we also have lipids. We build up, we make lipids. Lipids are at the top. So these are lipids, they are at the top. Who makes lipids for us? These are known as the free fatty acids, free fatty acids. We have the free fatty acids. We have the free fatty acids and glycerol. Don't forget glycerol. Free fatty acids and glycerol build up to give me lipids. Build up to give me lipids. So our lipids are my second form of macromolecules. I don't only have two, carbohydrates and lipids. What else do I have? I have proteins. So somebody said proteins are macromolecules. That's wrong. Proteins are macromolecules. So proteins are at the top. These are macromolecules. They are made up of micromolecules. Who are these micromolecules? These are the amino acids. So amino acids come together to give me macromolecule known as protein. Macromolecule known as the protein. Are we clear?
You said amino acid
Yeah.
increases.
Yeah, come together like building blocks to give me proteins.
Okay.
And lastly, we have nucleotides that come together, nucleotides come together in order to give me nucleic acids. Come together to give me nucleic acids. Come together to give me nucleic acids. Amino acids are not macro. Proteins. Proteins. Very good.
So and then nucleic acid, it's macro.
Yeah.
As well. Okay.
Yeah, and nucleotide is a micro. So this is going to make more sense when we look at biological molecules or large molecules built from smaller organic units. So you have carbohydrates, you see at the top, you see what my mouse is? The carbohydrates are at the top, then you have the lipids, then you have the proteins, then you have the nucleic acids. So they comprise most of a cell's dry mass. These biological molecules, they comprise most of the cell's mass, and they are all of them. This is very important. Nucleic acids, carbohydrates, lipids, and proteins, they all must have carbon. They all have carbon. And we don't say we all have carbon, we say they are all, we don't say they're all carbonated, we say they're all organic. All of them have carbon. Gotta have carbon. Proteins have carbon. Lipids have carbon. Nucleic acids have carbon, and carbohydrates have carbon. But in addition to the carbon, they may have other molecules. As we will see, the nucleic acids have phosphate. They have a phosphate group. Okay, now we said that water has very important properties. What are the very important properties of water? We said water is a versatile solvent. Water is a versatile solvent, meaning that it will dissolve most of the substance. And then water is very cohesive. So the water molecules stick to each other a lot because of the dashed line. Remember the dashed line from that Ball's model? That dashed line is known as what kind of bond is that? Yeah, so this dipole give you what kind of bond? Yeah, you're right, dipole. One is positive pole, positive pole, negative pole, double dipole. You're right. But what do we call? That's not the name of the bond. That's the description. Hydrogen bond. So this hydrogen bond make them stick to each other really well. Remember, I told you before that ammonia NH3, if you weigh it to oxygen H2O, are they different in mass? Are they different in mass? Yeah, they are different, but are they significantly different? Let me Google the, okay, molecular mass of ammonia. Can you guys see the molecular mass of ammonia? Ammonia has a N nitrogen and 3 hydrogens. What's the molecular mass of ammonia? 17 grams per mole. Please write that down. Molecular mass or molar mass of ammonia is 17 gram per mole. Now let's look at the boiling point or
Molecular or molar mass, 17 grams per mole.
yeah, and then let's look at the ammonia. What's the boiling point for ammonia?
25.
Please write that in Celsius. Minus negative, please write negative 33 degrees Celsius. If that temperature, ammonia will evaporate even in minus 33. Now let's look at water molar mass. Water molar mass. What is the molar mass for water? Yes, what's your question? You had your hand up. Yes.
23. You said it's 23? 33. Negative.
What is 30? The boiling point. The boiling point is minus 33. Minus 33. Now let's look at water. What's the molar mass for water? 18. Now let's look at the boiling point for water. What's the boiling point for water in Celsius? How much? So now compare ammonia and water. How different are their molar masses?
Not that different, like 1 point.
Not that different. But look at their difference in boiling point. How much heat does water take to boil? 100 degrees Celsius. How much heat does ammonia need to boil? So what does that tell you? That water will resist heat. Why? Because of the hydrogen bonding. Because the hydrogen bond, the dashed line, will hold all the water molecules to one another, make it very hard for them to evaporate. So it's not just about how heavy you are, 17 or 18, it's about this cohesiveness. So water is very cohesive. Water is cohesive due to the hydrogen bonding. Hydrogen bonding. In addition to water being cohesive, what else is so special about water? Water is adhesive. Adhesive. Water is adhesive. What does adhesion mean? It will stick to surfaces. If you put a paper slip, water is gonna climb up. If you put a piece of glass, water is gonna climb up the glass because glass is minus. Glass is minus charge, and water is gonna climb up. Okay? So also adhesion is important. That's why our lung has an adhesive serous, S-E. It has an adhesive serous fluid, has an adhesive serous watery fluid. It has an adhesive serous watery fluid. Does that make sense?
That's our lungs, right?
So that's a capsule around our lung.
Okay.
But if this capsule, so our lungs have a bag around them, and in between the two bags is water, because water will adhere to the lung to protect it. Also the heart, this is called the pleura. This serous fluid in the lung is called the pleura. There is another serous fluid around the heart, and that is known as the pericardium. The pleura is the lung. The pleura is for the lungs. And for the heart, it is known as the pericardium. Pericardium. Yum. Are we clear? Okay. Now let's move on. So, I'm sorry? Yeah, you do need to know.
Pericardium.
Yeah, you need to know an example of the adhesiveness of water to help you survive. The pericardium, that is the sac of water around the heart. The pleura, that is the sac of the water around the lungs. Everything I say in this class, you gotta know it, unless I'm saying a joke or something. But everything I explain all right, you gotta know that.
You said the pericardium is the sac around.
Cardium, not cardian, pericardium,
All right.
and then the pleura. Okay.
You said this is. Yeah.
Okay, that's recorded. I have to go. That's recorded. So you will see it. There's a lot to cover. The pleura is for the lung, and the pericardium is around the heart. So what were we doing now? Talking about cohesion, adhesion, solvation. That's showing you the property of water. Now, this is dehydration. That's another property. And hydrolysis is another property of water. We use water to split up. So this catabolism we talked about breaking down, we don't break down with a knife in our body. We don't break down the lipid with a knife. We don't break down the protein with knives. We break it down with water. So the enzymes utilize hydrolysis. And when you see lysis, when you see lysis means the breakdown. Lysis means the breakdown. Lysis means the breakdown. Lysis, breakdown. Hydrolysis is the breakdown. So that means if I want to break down, I add water to break down, to separate. So if I have two molecules, I add water, and I cause the lysis, the breakage, and the separation. Or I remove, minus. I take away water. That's known as dehydration. Dehydration.
Okay.
So you see there, what do we have? We have the dehydration synthesis. So if we examine now this particular, if we examine this here. That's so you don't need to know exactly what kind of sugar is that. So you have to look in what I'm pointing with my mouse. This is very important. I don't know how to get this out of the way. So this is dehydration synthesis. I remove water to synthesize. Remember we said monomers covalently bond to each other to create polymers. So when you go from monosaccharide to disaccharide to polysaccharide, that's very important because you use water removal. You remove water and then you bind them together. That's how you do it. Please look here. You see the OH red? These are all separate molecules. That's molecule number 1, molecule number 2. Do you see molecule number 1? Molecule number 2? Do you guys see molecule number 1 where my mouse is and molecule number 2? Now they are separated. But if you look here, they are coupled. So now they have been stuck together. They stuck together by removing water. So this molecule here will say, I'm going to get rid of an OH. And the other molecule will say, I'll get rid of an H. And in the end, you'll get plus H2O, meaning it left. And now they are all bounded by this oxygen that did not leave. So it's dehydration. You remove the water. That's very important. You build up by removing the water. That's why we call it dehydration synthesis. You synthesized what? You synthesized a dimer. You had a monomer, now you have a dimer. You had a monosaccharide, now you have a disaccharide, thanks to the dehydration. But let's say you want to break this disaccharide, because we don't just build up all the time. You build up when you have too much energy. When you have too much energy, when you have already too much energy, you're not burning it down, you build up. But let's say you started running. Now you need to burn down. So when you burn down, you need to break down this di into mono. So now you do the opposite. You add water, known as hydrolysis. So you put an OH back to this molecule and an H back to this molecule, and now they're separated. So this reaction here is reversible. It can build up by dehydration, and it can separate by hydrolysis. This is really important, and that shows you the properties of water, that it can break down. So it's not just the water is a versatile solvent, not just the water has high thermal capacity for sweating. It helps you to absorb the heat. Not just the water is adhesive in the pericardium and in the pleura. Not just the water is cohesive. It has a high boiling point. Water is also important for dehydration and for um, dehydration synthesis and hydrolysis. Very good. Now there you go. That's the breakage of the hydrolysis. Do you guys see the hydrolysis now? This is the opposite. Dehydration synthesis is the opposite of hydrolysis. So in dehydration, we removed water to bind them together. Hydrolysis, they are already bound, but we add water to them and then we separate them apart. Depends on the demand. If we need energy, we use hydrolysis to break them apart. If we don't need energy, we remove water to join them together. Now, you have to understand that these reactions will take thousands and thousands of years. If they're going to take thousands and thousands of years if we don't have an enzyme. So there are specific enzymes that catalyze this dehydration. And you know, enzymes don't work for free. Enzymes need ATP, need energy. And hydrolysis is energy releasing. So when you break down, so which one is dehydration anabolism or is dehydration catabolism?
Anabolism.
Yeah, dehydration is synthesis. You build things up, that's anabolism. Excellent, good job. And when we say hydrolysis, is that anabolism or catabolism? Catabolism. So which one is going to give me energy? Dehydration or hydrolysis?
Metabolism. So, what do you what?
Hydrolysis. And which one is going to cost me energy? Anabolism, because I'm stacking them together. So digestive enzymes, and you can tell digestive enzymes, they perform digestion. Digestive enzymes, they perform digestion. They digest the molecules, and they provide energy. So they digest, they break down. So that's hydrolysis. They hydrolyze. That's hydrolysis, and um, they hydrolyze and they release energy, providing energy for breaking them into smaller molecules that cells can absorb. You see, cells cannot absorb polysaccharides, disaccharides. They can absorb monosaccharides. So you see, that's the starch, and you see that is these are we break down starch into disaccharides. Then you use salivary amylase is going to be in your saliva. It's in your mouth, in your saliva, salivary amylase. Then there is another amylase in the pancreas known as pancreatic amylase that's going to further break down sugar into glucose.
So those are the enzymes.
Yeah.
Breaking down.
Yeah.
Food.
If you end with A's, not food, amylase just for sugar. For breaking down proteins in food, we use peptidase. We use peptidase. We cut down the peptides or proteases. For lipids, we use lipase. So always ending with A's, the enzyme with an A's. So the biological macromolecules are large and they are life essential, but they are built from smaller organic monomers. The four major macromolecules are proteins, carbohydrates, nucleic acids, and lipids. Monomers are joined by covalent bonds. They form polymers with new properties and lower osmotic pressure. Dehydration, condensation reactions join the monomers together and they release water. They release water. And hydrolysis breaks the polymers down using the water. You really need to know that. How dehydration works to remove water, to stick things together, and how hydrolysis works to add water to separate things apart. So dehydration reactions require energy input while hydrolysis releases energy. That's part one. 3.1. Now moving on to 3.2. Okay, if we go to 3.2, so carbohydrates are macromolecules with a ratio of carbon to hydrogen to oxygen. So that's the first class now. We're going to take them step by step. There's carbohydrates, there's proteins, there's lipids, there's nucleic acids. So now we're going to go through all of them. I'm sorry that there is no screen here, but you really need to be looking at that PowerPoint slide so that you understand.
Is this the same PowerPoint we got
On our computer.
Yeah,
on our computer?
yeah. So you can see my mouse pointing. I need you to see. Okay, it's up to you. I just want to bring your attention to the importance of following the PowerPoint slides. So carbohydrates, these are the sugars. These are macromolecules with a ratio of carbon to hydrogen to oxygen of 1 to 2 to 1 that are essential in our diet. So they providing energy through glucose, and they are present in natural sources like grains, fruits, and vegetables. So these carbohydrates, they are macromolecules. And remember, they must have 3 elements in this ratio: carbon, hydrogen, oxygen. There's always double carbon, hydrogen, oxygen. There's always double hydrogen. There's always twice as much hydrogen as there would be oxygen. So if there is 1 oxygen, there's gotta be 1 carbon and 2 hydrogens. That's the ratio that they work with. So this is the stoichiometric formula. So you see there's always 2 hydrogens compared to 1 carbon, 1 oxygen. But that does not mean all carbohydrates look like that. That's why there's an N outside. So if you have an N outside, let's say this N is a 2. So you multiply 2 by everything that's inside. 2 times C, that's 2C. 2 times 2H now, that's 4H. 2 times O, that's O2. So that's going to be C2H4O2. That's a carbohydrate. Every molecule they give you, if you can reduce it down to this stoichiometric formula, that means now you really got it. That's a real carbohydrate if it fits that formula. So that, remember the law is the ratio of carbon to hydrogen to oxygen in carbohydrate molecules is always 1 to 2 to 1. Glucose has the chemical, I'm sorry?
I actually get it. I didn't get what you were talking about, but I get it. Like when I look at the glucose formula, it's 6 carbon to 12 hydrogen to 6 oxygen. So that's the 6 is with the 1.
Yeah, but you got to take the N. So C6, that's it's very good that you see you observing well. If you remove, so now it's the C6H12 and O12. Who's that? That's glucose. Now, I'm sorry, O6. But remember, this is not the reduced, the formula says you must have a CH2O and an N on the outside. So now put glucose for me in this N formula. CH2O. What should the N be?
6.
The N is a 6. Excellent. Because 6 times C, that's C6. 6 times 2H, that's 12. And 6 times O, that's O6. Very good. You got it.
So how would you, what the end is?
You solve that. You divide, so you know that always everything here is, the hydrogen is always going to be double the oxygen and the carbon. So you divide by 2. You divide H12 by 2, you get H6. You divide O6 by 2, you get
3.
you no, this is oops. You, because you're gonna get CH2. So you're gonna divide by 6. Gonna divide by 6. So 12 divided by 6, that's gonna give you 2. Oxygen divided by 6, that's gonna give you 1. And carbon divided by 6, that's gonna give you 1. So to solve for the N, so the N just shows you there is a common number that you multiply all of the constituents in the stoichiometric formula for the carbohydrates, and you will get your carbohydrate of interest. But they must fulfill this law, CH2O. And it's easier to know because it's H2O, water, add a carbon to it, and then make the N outside. That is your law for carbohydrates. If it doesn't fit this formula, if you cannot break it down into this formula, it's not a carbohydrate. Gotta fulfill that formula. So glucose has the chemical formula C6H12 and O6. C6H12 and O6. So the term carbohydrate, the term carbohydrate originates from the components carbo and water, hydrate, carbo and hydrate. So scientists classify carbohydrates into three subtypes. We talked about that. Monosaccharides, disaccharides, and polysaccharides. Monosaccharides, who are my monosaccharides? When you see them, you gotta know these are my monosaccharides. These are my monosaccharides. There are three monosaccharides. The first one is glucose. Glucose is a monosaccharide. The second one is fructose. Fructose is a monosaccharide. And the last one is galactose. Galactose. That is a monosaccharide. Fructose, glucose, and galactose. Okay, guys. So fructose, glucose, and galactose. So fructose, glucose, galactose, those three represent for me my monosaccharides. Those three represent for me my monosaccharide. But I don't exist as a monosaccharide. I must become a disaccharide. So now I gotta join them together. So now let's mix and match. Glucose that binds with another glucose, that's gonna give me a disaccharide known as maltose. Maltose. That's the sugar for what? Maltose is the sugar for grain and oatmeal. That is grains, sugar, oatmeal, sugar. Are we clear?
So bring the sugar to maltose, what you said?
Yeah, maltose. And whose maltose is a disaccharide made up of two glucoses attached together. Then the second disaccharide is the sugar from the milk, lactose. Lactose. Lactose is made up of glucose, and galactose. Galactose. Students think because galactose has a longer name that it is the di, and lactose, because it has a shorter name, it is the mono. No, the galactose, the long one, is the monosaccharide, while the lactose is a double up, glucose and galactose. And the last type of disaccharides we are dealing with, maltose. Lactose, what is the last type? Sucrose. Sucrose is made up of glucose and fructose. Excellent. Glucose and fructose together give you sucrose. Sucrose. So what do you observe here in these three disaccharides? All of them gotta have what? Glucose. Glucose is part of every disaccharide out there. The second issue, glucose, when glucose is doubled up, when you have two glucoses attached together, what do we call that? What do we call the two glucoses attached together? A disaccharide, but what is the name of it? Is it maltose? That's maltose. That's maltose. And maltose is what kind of sugar? Is the sugar of the grains and the oatmeal. Okay, then you have lactose. So you know the law. Already you gotta have glucose. But glucose gotta bind who now? It's gonna bind galactose. It's gonna bind galactose. And lastly, you're gonna have glucose and fructose. Glucose and fructose, and that's gonna be glucose and fructose, and that's gonna, so this is lactose. Then you have glucose and fructose, and that's gonna give you sucrose. So what else do you notice? What about this bond in the middle? What's the name of this bond that connects two monosaccharides together? What's the name of this bond called? That's a hydrogen bond? G L Y C
Right.
O S. Somebody say glycerol. Are we dealing with lipids? Glycerol is for lipid. I D
Right.
I
Right.
C.
We can't even pronounce that.
Right. Acidic bond.
Yeah. You see? Kareem said it best. Shaquille. Shaquille said it best. Shaquille said it best. Oh my God. I had another Kareem. He's just like you. I'll get him, but he's big though. He's older. He's older. He was taking biochemistry.
I bring my hair, I want to see what you say I look like.
Okay, I'm gonna. You look like him in the way you act. His name is Kareem. I'll show you Kareem. Kareem was headache. Headache, big headache. You are, of course you are. Okay, all right, but you're good. But you're good. Both of you guys did excellent. Both of you guys are gonna do excellent. So, Shaquille, what is the name of this bond? That's gonna be on the exam.
Glycosidic bond.
Glycosidic bond. Yeah. Yes.
What does the glyphosate bond do?
It's a bond. So it's gonna connect to. It's gonna connect to. Excellent. Good job. Very good. Yeah, it can bind two glucose, combine glucose galactose, glucose fructose. That's a glycosidic bond. Okay?
Does the bond name changes for each monosaccharide?
No, it's a glycosidic bond. Okay. Glucose has the chemical formula, we've seen that, and carbohydrate, carbon, and you can tell from the name, carbohydrate, carbon and water, carbon and water. Monosaccharides. So monosaccharides are simple sugars such as glucose that typically contain 3 to 7 carbon atoms with the names ending in O's. Aldoses contain an aldehyde group. Aldoses contain aldehyde group, and ketoses contain a ketone group. Ketoses contain a ketone group. They can be trioses, 3 carbons, pentoses, 5 carbons, or hexoses, 6 carbons. All biologically relevant monosaccharides have a linear arrangement with a carbonyl. Remember the functional group we did last week? You got to know your functional groups. You got to know the carbonyl. This is going to go with you even to anatomy. You're going to take anatomy and physiology. Raise your hand if you're currently taking anatomy and physiology. So you should know. You should know that okay. So it's okay. So you need to, you can't forget it. You can't just take it and pass it and move on. You always got to go back to it. You always gonna go back to it. It's like something you keep going back to. This education is not something you leave behind. You're gonna go back all the time and read. That's how it's gonna stick. But if it's a one-time thing, you want to cross it, you're gonna forget it. It's gonna reflect bad in your profession. No, it's not. In the beginning, in the beginning, everything is hard. In the beginning, everything is a challenge. Running the marathon, the first marathon is the hardest. But then when you try, it's gonna become like running a mile. But you just have to keep at it though. You have to
You
keep
said
it.
you the A and P teacher, right?
Yeah. Okay.
On school?
Yeah. 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 I don't know what's happened. Only God knows tomorrow. I don't know nothing.
Good answer. Great answer.
Okay, I don't know. Nobody knows what's gonna happen tomorrow. So you see your this is a very nice, I like this depiction a lot. Because back when I was in college, I had a difficult time knowing the difference between the aldose and the ketos. If you look here, identify your carbonyl. Can you see your carbonyl where my mouse is? That's the carbonyl. So the carbonyl is attached to a carbon in one hand, a string of carbons, and a hydrogen. That's aldose, an aldehyde. But a ketone, look at a ketone, is attached to a carbon on the left arm has a carbon, the right arm has a carbon. But in aldose, the left arm has a carbon, the right here, this is the aldehyde. This is the carbonyl. The left arm for the carbonyl has a carbon, the right arm has a hydrogen. But in the ketone, the right arm has a carbon, the left arm has a carbon. Both arms are attached, are bonded with carbon. That's the ketone. So if a sugar is a ketose, it's gotta have a ketone. If a sugar is an aldose, it's gotta have a sorry? It's gotta have a carbon and a hydrogen. Very good. So do not think that it's just a one-time thing and you're done. No, you're gonna go back to it. You're gonna see the stuff we studied. You see today we're doing macromolecules and we talk about water. This was last chapter. We're gonna talk about it. So this is gonna go with you. You're never gonna leave something behind. You're gonna keep going with it. You see trioses? What are trioses? Trioses have, please look at my mouse. Trioses have one carbon, two carbon, three carbon, three carbons. That's a triose. Pentose has one carbon, two carbon, three carbon, four carbon, five carbon. That's a pentose. Hexose has a one carbon, two carbon, three carbon, four, five, six. That's six carbon. Are we clear?
No, because what started with
These are the names. Pentagon is five. What is it?
well, how much carbon does the carbon look, the aldose looks like it has three carbons? Yeah.
No, this is a different classification. This is a classification on the right here. When you look at triose, pentose, hexose, that's how many number of carbons do you have. Pentagon is a five. Tri is a three. But aldose and ketose is the carbonyl attached to two carbons, or is it attached to one carbon and a hydrogen? Like this triose is a aldose because it has three carbons and the carbonyl has a hydrogen. Do you get it? So these are two separate classifications. One is based on the quantity, the number of carbon atoms, and the other one is based not on the quantity, is based on the structural classification of the carbonyl. Is it aldose or is it ketose? So just recognize your carbonyl. If it's attached to an H, that's an aldose. If it's attached to one carbon, one H, that's aldose. One carbon, that's a ketose. Are we clear? Monosaccharides. So in humans, glucose is an important energy source that helps produce ATP during cellular respiration. So in humans, glucose is an important energy source that helps to produce ATP during cellular respiration. So you see that we catabolize, we break down glucose, we catabolize, we break down glucose in order to get ATP. So plants synthesize glucose during photosynthesis and they assemble it into starch, and humans obtain glucose by breaking down this starch. Are we clear? Galactose, fructose, and glucose are isomers with the same chemical formula, but they have different structural arrangements of functional groups. You see, galactose, fructose, and this is glucose. So glucose, fructose, and galactose, do they have different numbers of carbon? Do they have different numbers of carbon? No, they have the same number of carbon. They have the same number of hydrogen, the same number of oxygen, but they arranged differently. So look at glucose. How many carbons does it have? One. That's here in this intersection. That's a carbon. So one carbon, two carbon, three carbon, four carbon, five carbon, six carbon. That's six carbons. Let's look at fructose. One carbon, two carbon, three carbon, four carbon, five carbon, six carbon. And then you have one carbon, two carbon, three carbon, four carbon, five carbon, six carbon. Make sense? So they all have the same number. They are arranged differently. It's like 1, 2, 3, 4, 5. Then you put 1, 4, 5, 3, 2. Then you put 2, 4, 5, 3, 2. It's like that. So you just rearrange, but they have the same number of atoms. Monosaccharides can form linear chains or ring forms. So these monosaccharides, they can become cyclic, they can cyclicize, and they can be like a string. So they favor the ring forms in aqueous solution. So when you put a monosaccharide in an aqueous solution, it's going to be a ring form. It's going to be a ring form. So the ring formation makes the carbon at the end of the chain lose its double bond to oxygen to complete its attachment to the ring. To complete its attachment to the ring. The hydroxyl group on carbon 1 can be alpha or can be beta above the ring. So these orientations create structural isomers of monosaccharides. They create structural isomers. So you see here, this is a conversion. This is an interconversion between a ring and this is the linear form. You see the linear form? It will undergo interconversion to give you these two structures. So you see, the hydroxyl group on carbon 1 can be alpha below the ring or beta above the ring. You see, this is the alpha group. If this hydroxyl group here, if it's above the ring, that makes it an alpha. If it's below the ring, that makes it a beta right here. So you see, the hydroxyl group on carbon 1, where is carbon 1? This is carbon 1. This is carbon 1. If the hydroxyl group is above the ring, hydroxyl group on carbon 1, if it's below the ring, that makes it alpha. If it's above the ring, that makes it beta. Where is carbon 1? That's carbon 1. So it's going to cyclicize. This is my carbon 1. If my OH is below the ring, like here, that's alpha glucose. If the OH is above on the top, that's a beta glucose. And these are my disaccharides. Remember my disaccharides? Di means two. They form via dehydration. So I remove water to link them together. You see, I removed water to link them together. We talked about that. Who does glucose and fructose come together? Please, do you see where my mouse is? Glucose and fructose come together to give me sucrose. Is that clear? So the glycosidic bond is alpha when the carbon 1 OH group is below the ring, and beta when the OH is above the ring. So that shows you, and what's the name of the bond? Glycosidic bond. When it's connecting an alpha carbon, when the carbon hydroxyl group is below the ring, like here. When the carbon is above the ring, that makes it an alpha. If it's below the ring, that makes it a beta. Yes.
The 6 and 6 makes 12 for the sucrose.
Yeah, because they're all gonna be bonded as one molecule now.
Well, why is the oxygen one 11 instead of 12?
Because remember in the dehydration, we're gonna remove water in the dehydration. So it's called dehydration synthesis. I'm gonna stick them together by removing water. So you see here, I removed an OH here and an H here. So the water came out here for them to stick together and make a glycosidic bond. Make sense? The water has to come out. Like we said this earlier in the other, yeah. In order to make this bond, this glycosidic bond, you have to eliminate OH from here and an H from here, H2O, so water can come out. That's why we have dehydration, removal of water.
Okay.
So when then they're gonna stick like that. So this glycosidic bond is gonna become an alpha glycosidic bond when the carbon hydroxyl group is below the ring, and the beta when the carbon hydroxyl group is above the ring plane. So let's look at sucrose. In sucrose, that's an alpha. So we talked about that. That's a very important slide. These are the common disaccharides. You have lactose. Milk is made up of glucose and galactose. Maltose is made up of glucose and glucose. Good. Sucrose is made up of glucose and... Now, a polysaccharide is a long chain of monomers linked together by what kind of bond? By a glycosidic bond. It can be an unbranched, linear, or branched glycosidic bond. Branches are strengthened by the hydrogen bonds, and examples include starch, glycogen, cellulose, and chitin. What's chitin? What's chitin? Ever heard of chitin?
Is it fat?
Fat?
Is it?
You know, in chitlin.
That's what.
Anybody before coming into this program did bachelor's degree? Anybody? You did? Oh wow! In what, Mahanur? In psychology, biology, and you did it in what? Psychology and natural science. So bachelor's. So did you guys take biolab in biology a long time ago? So do you remember chitin? Chitin. So chitin is in the exoskeleton of the chitin is found in the exoskeleton of the crustaceans. In the the crab. The crab and the shrimp. Yeah, their skin is chitin. So that's a special type of sugar.
Is it like plastic?
Yeah. I'm sorry? Yeah, that's chitin. Yeah. Also on the surface of the cockroach. Yeah, it has chitin. That's chitin. So are you guys going to do the BSN program? When you finish the LPN, you're gonna go do BSN, bachelor's? I'm sorry? This is the bachelor's in nursing? So you're not LPNs? Oh, so you're gonna have bachelor's. Oh wow, very good. That's good. So in how long is gonna take? So this is your first semester.
Yeah.
How many more semesters and you'll have bachelor's in nursing?
About
2.5 years, 5 semesters? But the semester is like 3 months. 8 semesters? How many semesters is this thing?
It's
And
28 months, I think. I don't know.
8 semesters, and the school has how many? School has 4 semesters, right? Yeah, so 2 years. Okay, so if it's 8 semesters, this year has 4 semesters. January to April, then we're gonna begin end of April to September. Then oh yeah, 3. 3, yeah. Okay, no problem, but it's good. You're gonna have a bachelor's now. And you're gonna have double bachelors. Very good. You two over there. You 3 over there with Navraj.
How long is the break?
The break? The break is like a week.
Eastern.
Yeah.
So what about the summer?
Or like 10 days. That's good for you, yeah. So okay. So that means I'm gonna see you for pathophysiology. I'm gonna see you for biochemistry. Oh my God, yeah. So that's good. That's the best program to have bachelor. It's gonna help you a lot. That's really good. Yeah, I'm happy for you guys. I want all the LPN to become bachelor. That's very good. Very good, guys. Good for you. Good for you. Yeah, I would have done it in a heartbeat. In 2 years, you'll be done. 2 years and a half. 2.5. That's okay. Yeah, that's really good. And you're gonna graduate as RNs. Shaquille is gonna become the RN. Yeah. Yes, my man Shaquille. Yeah. He's not gonna give me painkillers. What? Oh no, my bad. Yeah. Shaquille's gonna say, listen, that guy don't give me painkiller. Just leave him. He gave us too many assignments.
I'm not gonna do that to you. Make sure you get help.
He'll do something else. Okay. All right, guys. I'm so happy. You're gonna do bachelor's. Excellent. I want my friend to do it, but he doesn't want. He watches anime. My friend, I want him to do it. He doesn't do it. He doesn't listen. He doesn't do anything. He doesn't care. Okay. Anyways, so polysaccharides. So remember. So now working our way up. We went up from monosaccharides to disaccharides to polysaccharide. One example of polysaccharide is talked about chitin. Then you have starch and cellulose. So the first one is starch. That's your storage sugar, and you store the sugar in your body in the form of starch. That's very important to know. So polymers that store excess glucose storage in plants, such as amylose. This is unbranched glucose polymer, and amylopectin. This is the branched glucose polymer. So they're stored in the roots and the seeds of the plants. So enzymes like amylase in the saliva. So remember, that's why we call it salivary amylase. I don't like it when they say that. They should come together. Salivary amylase, so it sticks to your mind. It's in the saliva. It comes out in the mouth first. And they break down starch into smaller molecules like maltose and glucose for cellular respiration. Glucose monomers in starch are joined by alpha 1 to 4 or alpha 1 to 6 glycosidic bond. Okay, this is the amylose, and this is, you see now the hydroxyl is on, so this is an alpha group. You see how amylose, what's the difference between amylose and amylopectin? We said in the last slide, one is branched, one is unbranched. Please look here, look where my mouse is. Amylose is unbranched, long linear polymer chain, and amylopectin is branched glucose polymer, stored in the roots and the seeds. So you see, you have amylose, and then you have amylopectin that is branched. Then you have polysaccharides, glycogen, and cellulose. In cellulose, glucose monomers are linked in unbranched chains by a beta 1,4 glycosidic linkage. By a beta 1,4 glycosidic linkage. So glycogen is branch polymer for glucose storage in animals. So glycogenolysis, from the name, lysis, glycogenolysis. So we lyse glucose. Is that anabolism or catabolism? Glycogenolysis, catabolism. They break the breakdown of glycogen to release glucose into the bloodstream. You see, we're asking, this is not nothing personal to nobody. You know, back when I was in college, I didn't do good because I didn't study the traditional way. I want to learn and understand. So I used to spend so much time reading the book, reading the book, reading the book, but the semester would pass by so quick and I wouldn't catch up because I can't read the whole book in a very short time. Now my friends, they just scam and cram through and they just pass. But in the end, when I became a professor, all the time I spent to learn helped me a lot. Now I'm asking really basic questions, and you see some students, they took the classes. They have you guys have bachelors in biological and natural and psychology, but you don't remember very basic. So I feel it's the way that the courses are taught and how students are required to learn. It's just you just need to go through. This is really bad. You have to learn. You have to read the book. You have to sit down and open the book and be patient. You know, they work out in the gym. You have to work out your mind. You have to be patient. And you read the sentence. The first time is not going to make sense. You read it the second time, the third time, the fourth time. Then you keep reading the chapter. Read. Then you will have a deep understanding. Otherwise, you're going to store everything in short-term memory and you're just going to be cramming through. You're passing your exam, but you don't understand anything. You have the degree, but really you don't understand. So you have to get the best worth of your education. If you had a degree in the past, now you're starting another chance, don't repeat the same mistake. Please invest more time. Invest more time in reading, writing, learning more. Don't look at it like, oh, this is just a class I need to pass. No. Think of it as something you need to take home. You take it in your mind. You take it in your heart. Keep reading and learning. You will see it again. I guarantee I teach, like I told you, A and P. This is all A and P. You will see it again. So now you're going to do double job. You have to be businessmen and businesswomen. You have to be a good investor. So you invest once and for all. You learn what glycogenolysis is. When you go again in anatomy and physiology, it's going to be like a you're listening to a story again. But if you don't do it now, you're gonna defer it for later. So yeah, no problem. So please invest and read. This is the Hawkins learning. You paid for that. Please read, do the assignments, and I want everybody to learn, everybody to get an A. How did everybody do on exam today? So okay, so now that's gonna help with that first time. But next midterm, there is no midterm B, so you have to be very careful. You have to do your homework and you have to read the review the exam questions and you have to review the homework questions I'm gonna post for you on Canvas, and you're gonna review the Hawks and the study guide. And then you're gonna come next week and you're gonna take your midterm because we don't want to be behind. Yes.
For exam B, are you still gonna get credit for the, yeah, for the study guide?
What do you mean?
The one that we did.
There's one study guide for both A and B. That you just do that, you get your full credit. But you have to submit it. Make sure if you have like an exclamation mark or Trinity didn't scan it, it's still open until I believe.
Still gonna count towards the test, right?
Of course. So you have February, it's due until February 9th. So okay, go work on that and make sure your plagiarism checker does not do 30. Beyond that, the next study guide for the midterm, it has to be 25, guys. Cannot be more than 25. Okay? Yeah, we said this last week.
Dropping the study back for the midterm.
Yeah, that's gonna be Thursday. So you need to go study. Yeah, you need to go read. Yeah, you gotta go read the chapter first because I give you the study guide. You're just gonna go read the study guide question and just focus on that. No, I know you need to read the whole chapter first in the Hawks. You have to read the whole chapter, and the PowerPoint slides is all based on the PowerPoint slides. No, 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 no. We cannot be behind. We cannot be behind. No, 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 no. I cannot do that. I cannot, guys, please. So please read the PowerPoint slides. All of that. The study guide is not going to ask you about something in another book. It's all this. So you got to go over all of that. You're paying attention now. It's going to help you a lot. And then you go home, you review that.
What chapter is it gonna be based on? It's based on the chapter that.
Everything. Chapter 1, 2, 3 today, and 4 today. The 4 of them are going to be on the midterm. So you will see that.
Based on the exam that we did,
It's going to be based on all the information.
are you gonna add more?
Of course.
Oh.
Okay. Yes. You got to look at the syllabus. Yeah, you got to do that. Okay. Glycogen. Glycogen is an example of a branch polymer for glucose storage in the animals. So we store glucose in the animals in the form of glycogen. We break it down in glycogenolysis. So you know that glycogenolysis from the name lysis, we break down glycogen to get energy. Now cellulose is a biopolymer that forms rigid plant cell walls. So cellulose is in the plants. Humans cannot break down cellulose, but herbivores. What are herbivores? Yeah, like the cow.
Yeah.
The cow, they have the. Why can't we not break down cellulose? Because in cellulose, the glucose monomers are linked in unbranched chain by a beta 1,4 glycosidic bond. We can only break down alpha 1,4 glycosidic bond. Does that make sense? Chitin, you see, the exoskeleton is a nitrogen-containing polysaccharide that form exoskeletons of the arthropods, like the insects and crustaceans, and a major component of the fungal cell wall composed of repeating N-acetyl B&D glucosamine NAG units. Just remember that chitin is a nitrogen-containing polysaccharide found at the surface of the exoskeleton of the insects. Benefits of carbohydrates for the overall health. So they provide energy through glucose breakdown and ATP production. They contain fiber, which aids in digestion and in cholesterol regulation. They aid in the digestion and in the cholesterol regulation. They have an important the importance they have been a part of a human diet for many years, and they are so this is the summary for carbohydrates, classified as monosaccharides, disaccharides, polysaccharides. We talked about all of that. Okay. So now we finished with one macromolecule, the sugars. Now we're going to move into the lipids. Lipids are non-polar, so they don't dissolve in water. They are hydrophobic compounds composed of hydrocarbon chains, long hydrocarbon chains. Hydrophobic lipids in the aquatic mammals first, such as the fur of this river otter. I hope I don't meet it in person. Protect the mammals from the elements. Have you ever seen this animal? An otter. Yeah, like a little, yeah. You see, all mammals have hair. Lipids are nonpolar hydrophobic compounds composed of hydrocarbon chains. They serve as energy reserves, insulation, and structural components in the cells. The major types include fats, oils, wax, phospholipids, and steroids. So that's the structure. I remember I was saying glycerol earlier. When I was writing the bond, glycosidic, I put somebody said glycerol. No. But look, the law is the same. When I want to hook them up together, I remove one hydrogen from here and an OH from here, and I end up with two hydrogens and one O. What does that sound like? Dehydration. I remove water and then I hook them up together. But who am I hooking up together? Glycerol and fatty acids. That's very important. So how many fatty acids am I going to have? 3. 3 is going to bind each carbon. So this long fatty acid is going to bind this carbon. This second long fatty acid is going to bind this carbon. This third long fatty acid is going to bind this carbon. So the 3 of them are going to now be forming a triacyl glycerol. Now you know why we call it tri? Like 3 of them. Triacyl glycerol. They get stuck together. So glycerol is going to be stuck like that. Does it make sense? So the fat molecules consist of glycerol and fatty acids. Glycerol is a 3-carbon alcohol with 3 hydroxyl groups. Fatty acids are long hydrocarbon chains with a carboxyl group, linked to glycerol by an ester bond. You see, this is the ester bond. Ester bond is an oxygen that has been connected to 2 carbons. Oxygen in the middle of 2 carbons. Oxygen in the middle of 2 carbons. So please remember that. I make my lipids based on 2 components. A glycerol. How many carbons does glycerol have?
3.
1, 2, 3. Excellent. How many fatty acids are going to bind to the glycerol?
21.
How many fatty acids are going to be hooked to the glycerol? 3. 1 long up here, the second long in the middle, the third long in the bottom. The same way, I'm going to remove an OH from the fatty acids, and I'm going to remove an H from one of the OHs of the glycerol. And then I'm going to make my dehydration synthesis to make this ester bond. What's the name of this bond right here? That's an ester bond. Is that clear? Now we have, when we hook them together, triacyl glycerol. When we separate them, we have glycerol and fatty acid. Triacyl glycerols are fats consisting of 3 fatty acids linked to a glycerol molecule, created by dehydration synthesis, which releases 3 water molecules. Fatty acids with single carbon-carbon bonds in hydrocarbon chain. These are saturated. So meaning saturated fatty acids, meaning they are saturated fully. All the carbons are full of hydrogen. There is no double bond. Unsaturated fatty acids, these are fatty acids containing double bonds between carbon atoms in the hydrocarbon chains. They are kinked chains that cannot pack closely together, resulting in a liquid state at room temperature. So examples are the olive and the canola oil. So these are unsaturated. You see there's a double bond. If there's a double bond, that is unsaturated fatty acid. So unsaturated fats are in either a cis or a trans configuration. So cis hydrogens are on the same plane causing a kink, while trans hydrogens are on different planes. You see, cis plane, this is cis meaning both are on the same side, and trans meaning that they are on the opposite sides. So trans fats are formed artificially by hydrogenating oils, leading to a trans configuration that does not naturally occur. So margarine and some peanut butters and the shortening linked to higher low density lipoprotein levels or bad cholesterol. These are the trans fats. So the deposition of plaque within the arteries is partially due to an intake of trans fats and causes a reduction of blood flow to that tissue. So one way to address this is to insert a graft which reroutes blood flow around the blood vessel. We have omega fats. Omega fats are polyunsaturated fats that the body requires but does not synthesize. These are polyunsaturated. So omega-3 fats in the fish have a double bond connecting the third and the fourth carbons from the omega end. That's the farthest end from the carboxyl group. That's the carboxyl group. That's the omega group. These are good dietary sources like the salmon and the plant oils. Wax is made of long fatty acid chains esterified to long chain alcohols. They coat the feathers of aquatic birds and some leaves. Hydrophobic nature prevents the water from sticking to these surfaces. Now, when we look at phospholipids, phospholipids consist of fatty acid chains attached to a glycerol or a sphingosine. That's an amino alcohol backbone. So they amphipathic. So phospholipids are amphipathic because they contain a hydrophobic core and a hydrophilic phosphate part. So they make up the plasma membrane mostly in the form of phosphatidylcholine and phosphatidylserine. So you see, this is the head group, this is phosphate, and this is glycerol. You see, now you see why the plasma membrane is called amphipathic. Why is the plasma membrane called amphipathic? Because it has a hybrid, 2-in-1. The heads are polar because of the phosphate group. You see the phosphate group? But the tails are hydrophobic. So if you see, what is this here? These 3 carbons, carbon, carbon, carbon. What is that? That's the glycerol. That is a glycerol. The only difference is it still has the long fatty chain on one, the long fatty chain in the middle, but the long fatty chain in the beginning was cut and replaced by a phosphate. So it's a glycerol that has one fatty acid head cut off and a phosphate was put there. So now we can have a 2-in-1, a polar hydrophilic head and a non-polar tail. Do you get it? So this is my glycerol that has a phosphate head and two tails that are hydrophobic. And you have double layer of that. So that makes a amphipathic bilayer. There's a double layer. They make up the plasma membrane in the form of phosphatidylcholine, and you have phospholipids or amphipathic. They contain a hydrophobic tail and a hydrophilic head. Amphipathic mean that they are a hybrid, two in one, polar and nonpolar at the same time. But who's polar? The head. Who's nonpolar? The two tails. But you have to know that the phospholipid, what is this called here? That's the phospholipid because it has a glycerol that's connected to two fatty chains as normally, but the head is no longer having this tail, and we replaced it with a phosphate. Let's look at the structure and the properties of the phospholipid bilayer. These are our plasma membranes. You see they form the phospholipid bilayer. You see that's the hydrophilic head on the top and on the bottom. So you have double layer. So this layer is doubled. The top you have phosphate heads and tails, and on the bottom you have phosphate heads and tails. On the bottom you're facing the aqueous cytoplasm. The cytoplasm is a colloid, an aqueous colloid cytoplasm. And on the top you have an aqueous, on the top you have an aqueous, on the top you have an aqueous, a phospholipid head. So you see, so you're facing the extracellular space that is polar, so that's facing the phospholipid bilayer. On the bottom you have the cytoplasm that is facing this phospholipid bilayer. So do you see that? Do you see the amphipathic? And there is double. So this is amphipathic, meaning one has a phospholipid head, hydrophilic, and a tail, hydrophobic. So that's itself amphipathic. But you have double amphipathic. You have double bilayer. That's why they call it bilayer. Two. Bilayer. One layer at the top of amphipathic phospholipid and a layer on the bottom. Steroid. Now, if we look at the steroids, these are made up of cholesterol. If you see four rings, 1, 2, 3, 4, that's a cholesterol. The steroid is a lipid with four fused hydrocarbon rings forming a planar structure. So the hydrophobic and insoluble in water. So some, such as cholesterol, also have a short hydrocarbon tail. Okay, steroids. Many steroids have an OH functional group, classifying them as sterols, such as estrogen and testosterone. These are the sterols. Cholesterol is the most common steroid synthesized by the liver and is a precursor for steroid hormones, the vitamin D, and the bile salts. Sterols like cholesterol and the phytosterols are components of the phospholipid bilayer. So remember that cholesterol is an integral part of your plasma membrane. It adds to the structural rigidity, the strength of your plasma bilayer leaflet. It is cholesterol. What's the more cholesterol there is in your plasma membrane, the thicker and the stronger it is. Yes, I had a question in the back? So your hand up. Okay. So lipids are nonpolar. You got to remember that they are nonpolar, hydrophobic. Macromolecules, including fats, oils, waxes, phospholipids, and steroids. Fats like triglycerides, they store energy and are made of fatty acids and either glycerol or sphingosine. Fatty acids can be saturated with single bonds or unsaturated with double bonds in their hydrocarbon chains. And phospholipids form membranes and they have a glycerol or sphingosine backbone that is attached to two fatty acid tails and phosphate head group. Okay, we're done with the lipids. Who is our fourth macromolecule? The protein. I'm sorry? Who's the fourth macromolecule? Our fourth macromolecule is our protein. So proteins have different functions. Some of them are enzymes, some act as toxins, and some as channels. They allow for the movement inside and outside, the influx of ions, the efflux of ions. Proteins are constructed from amino acid polymers. So you see, we make proteins from amino acid polymers. Types and functions of proteins. So the first type of protein is the enzyme. Enzymes are catalytic proteins that increase the rate of biochemical reactions. Enzymes are catalytic proteins that increase the rate of biochemical reactions. They accelerate the rate of the biochemical reactions. They are catabolic enzymes that break down the substrates while the anabolic ones build complex molecules. You know that. Catabolic enzymes catalyze, speed up, break down of macromolecules into micromolecules. Anabolic means that they build up. They accelerate the building up of macromolecules. So hormones like insulin are chemical signaling molecules that regulate physiological processes. So changes in temperature, pH, and exposure to chemicals can lead to the denaturation and the breakdown and the falling apart of the proteins. So this table here shows you that proteins exist in a variety of types and they perform several functions. So there are digestive enzymes like amylase, lipase, pepsin, and trypsin. They help break down the food by catabolizing the nutrients into monomeric units. They transport oxygen and they transport other substance like hemoglobin transports oxygen and carbon dioxide, while albumin transports nonpolar substance that is dissolved in aqueous solution. So in order to we need a boat. We cannot swim in the river. So some nonpolar substance that has to be transported in our blood. If some nonpolar substance has to be transported in our blood, that means we're going to need a carrier. An example of that is going to be albumin. There are structural proteins like actin, myosin. These are important for skeletal muscle movement. Keratin is important for the hair and collagen for the nails. There are important protein hormones, catecholamines, like the insulin, like the thyroxine and the insulin hormones. Also for defense, you need your antibodies and your antigens. These are proteins. And we talked about actin and myosin. They are important for the contraction of the muscle cell. Storage. We have the legume storage proteins and egg white. These are there to store proteins, and they provide nourishment in the early embryo. These are the legume storage proteins and for development and in the seedlings. Okay, these are the amino acids. Amino acids are monomers that comprise proteins. So each amino acid has a central carbon bonded to an amino group, a carboxyl group, a hydrogen group, and an R group. That's an R group, amino group, hydrogen group, and a carboxyl group. They are named after the amino and a carboxyl acid group. So the R group or the side chain distinguishes each amino acid. There are 20 common amino acids found in the proteins. So you need to understand the structure of the amino acid. There's an alpha carbon in the middle. This is the central alpha carbon. It has 4 terminals: north, south, east, and west. So you have one hydrogen, then you have a carbonyl, or known as the carboxyl group, then you have the amino group, then you have the side chain R group. All amino acids have the same amino group, they have the same hydrogen group, they have the same carboxyl group, they just have a different side chain. The side chain is different. So if you look here, you see that's the carbon group, that's the carboxyl, hydrogen, amino. You see? Carboxyl, hydrogen, amino. Carboxyl, hydrogen, amino. Carboxyl, hydrogen, amino. Is all the same. The only difference is this R group. Is this R group right here. This R group is what makes all the amino acids different. This R group, this R group, this R group, this R group, this R group. That's what makes them different. Not the carboxyl group, all of them have it. Not the amino group, all of them have it. Not the hydrogen group, all of them have it. It is the R group that's different that makes all the amino acids different. This concept is very important. Now this R group, it could be polar and it could be non-polar. So if the R group is polar, that means the whole amino acid is polar. If the R group is non-polar, that means the whole amino acid is non-polar. Some have polar and hydrophilic side chains, like the serine, the threonine, and the cysteine. These are polar because you see they have the hydrogen. It can form hydrogen bonding. It can form hydrogen bonding. And some amino acids have negatively charged side chains. You see, aspartate has a negatively charged side chain. Glutamate has a negatively charged side chain. Glutamate and aspartate have carboxyl groups that can lose a proton, giving the side chain a negative charge. Other amino acids have positively charged basic side chains, like lysine and arginine. They have amine groups that can accept protons, making the side chains positively charged. There you go. You guys need to know this, that in every amino acid, you have a central alpha carbon. Now we're going to see how the peptide bonds are made. You're going to have a central alpha carbon, hydrogen terminal, amino terminal, carboxyl terminal. Is the R terminal that's different. Now, how do we connect them? How do we connect? We connect with dehydration. We remove an OH from here. Are you paying attention? We remove an OH from here and an H from here. What's the OH plus H going to give you? H2O. Then you make the bond. You see? So the carbonyl is going to donate an OH. The amino is going to donate an H. Then you're going to have a bond here. That's the peptide bond. The peptide bond happens when you remove water between two amino acids. Glycosidic bonds happen when you remove water between two sugars. So you're always removing water. Dehydration synthesis. But when you do dehydration synthesis for the amino acids, you're going to end up, you see water came out. You see where my mouse is? The water has come out because I removed here. I removed the OH from here. I removed the H from here. Then I connected the bond. Very important. Are we clear? So the name of the bond between the sugars is called a glycosidic bond, and the name of the bond between the proteins is called a amino group. Cytochrome C. Cytochrome C is a crucial protein. I'm sorry, please close the door. I don't want my voice all in the hallway. Thank you. Cytochrome C is a crucial protein in the electron transport chain containing a heme group with iron that is reduced and oxidized during electron transfer. So cytochrome C has high amino acid sequence homology or similarity across the species, indicating its importance and common ancestry. The human cytochrome C contains 104 amino acids, 37 of which are the same position as the other species. Now, when we look at the polypeptide structure, polypeptide is a chain of linked amino acids with distinct terminals. So there's a free amino group at one end, the N terminal, and a free carboxyl group. We talked about that. That's the amino group, the NH2, the green one. Then the carboxyl group is the red group. So after the synthesis, polypeptides undergo post-translational modification like cleavage or phosphorylation. So often they have bound prosthetic groups. These are non-peptide groups needed for biological function of the protein. So they fold into distinct three-dimensional shape essential for their biological function. So a protein shape or conformation is crucial for its function. So the enzyme must have an active site, and it must be the proper shape like a lock and a key to bind its specific substrate and catalyze the reaction. So an enzyme is never going to bind a different substrate. It must bind the same substrate it was intended to catalyze the reaction for. That door there, if you put in a different key, is never going to open. But if you have the same copy of the key, that's how we have the drugs, the pharmacological interventions. When you make a medication that is similar to a hormone in your body, it's still going to work because it looks just like it. It's going to fit the proper conformation, like a lock and a key. So alterations in the active site or the overall protein structure can prevent the proper substrate binding. And there are 4 levels of protein structure. There's the primary, secondary, tertiary, and quaternary structure. A protein's primary structure is its unique sequence of amino acids in the polypeptide chain. Mutations result in the wrong amino acid being placed within the chain. So you see mutations result in the wrong amino acid being placed in the chain. And that's what's going to happen with sickle cell anemia. In sickle cell anemia, you're supposed to have an amino acid known as glutamic acid. Glutamic acid. But a mutation happened, and therefore you're never going to have glutamic acid. You're going to end up with valine. And that's what's going to lead the cell to sickle. So the protein will come out as a primary structure, like a linear chain, long sequence of amino acids. And mutations result in the wrong amino acid being placed within the chain. Oh, we just said that. So sickle cell anemia results from a single amino acid substitution in the hemoglobin B chain, in the B chain. So the hemoglobin forms long fibers, distorting red blood cells into sickle shapes that clog the blood vessels. And complications include hypoxemia, loss of oxygen saturation, breathlessness, dizziness, headache, and abdominal pain, and so on. Now, when we look at the primary structure of the amino acid, that is the primary structure of the amino acid. This is the secondary and the tertiary structure, then a quaternary structure, and then some proteins, they work in a secondary structure. Like who? Who are the proteins that when they are secondary, they can work? You know, some people, they can get a job with a high school degree. They don't need college. Some people, they gotta have a bachelor's to get the job. Other people, they gotta have masters. So for example, some proteins, they need to be secondary. That's it. Like collagen, keratin, that's secondary. Only they don't need to be tertiary or quaternary. Just secondary, they can do their job. Collagen and keratin. Who's the tertiary protein that does its job as a tertiary protein? Myoglobin. Myoglobin. That's the muscle protein that stores oxygen in the muscle. And lastly, you have hemoglobin. Now that's got to be quaternary. Four subunits. Two of each, two alpha, two beta. So that's the secondary structure. You see, secondary structure is like an alpha helix. And we rely on the hydrogen bonding. You see, we never left hydrogen bonding. It's going to carry on with us. So if you roll this, remember we said the primary structure is like strings on a bead, like a train, like a long linear chain. If you roll this long linear chain on its axis, you get a helix that's known as an alpha helix. You guys see the alpha helix structure? But if you don't roll it and you plate it like a beta pleated sheet, that's going to be known as a beta pleated sheet structure. So the local folding of the polypeptide causes secondary structure of the protein. So most common structures are alpha helices and beta pleated sheets. They are stabilized by the amino acid hydrogen bonds. Alpha helix is a hydrogen bond between the amino acid, and it twists the polypeptide into a helical coil. There are 3.6 amino acid residues per helical turn. Now, secondary structure, an example of a secondary structure are the beta pleated sheets. The beta pleated sheets consist of linear beta strands that are connected laterally. Strands can be parallel or anti-parallel to each other. Just remember that beta pleated sheet and alpha helix are what type of structure? What type of protein structure? Alpha helix and beta pleated sheet are examples of what kind of protein structure? Secondary. And a long string on a bead, like that, long string on a bead, like a train. That's an example of what structure? Primary. Alzheimer's disease is believed to be caused by deposition of amyloid plaques in specific regions of the brain. Amyloid plaques are made up of beta amyloid segments that are sticky because they are made of short amino acid chains that arrange themselves in a secondary structure of beta sheets. The tertiary structure is a protein's unique three-dimensional shape caused primarily by the R group interactions. So you have hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bridges. So these are the types of bonds that help us to roll and fold up even more than in the secondary structure. That's the tertiary structure. Hydrogen bond, ionic bonds, hydrophobic interactions, and disulfide bridges that contort the polypeptide chain. The hydrophobic R groups interact on the inside of the protein with the hydrophilic R groups on the outside. There's a loss of tertiary structure can make a protein non-functional. So how do we end up with a tertiary structure? Through hydrogen bonding, ionic bonding, hydrophobic interactions, and these disulfide bridges. Quaternary structure is when you have multiple polypeptide subunits that have a quaternary structure made up of interactions between the subunits. So there are weak interactions that stabilize the overall structure. The insulin's two subunits are held together by hydrogen and disulfide bonds. So that shows you the primary structure. You guys see the primary structure? The sequence of a chain of amino acids. Then you have secondary protein structure. There's the hydrogen bonding of the peptide backbone. It causes the amino acids to fold into repeating patterns. Then you have quaternary protein structure. These are proteins consisting of more than one amino acid chain. And then you have the tertiary protein structure. These are three-dimensional folding patterns of a protein due to the side chain interactions. These are the tertiary structures. So if you increase the temperature too high, what's going to happen to the protein? It's going to denature and fall apart. So environmental changes like the temperature, the pH, and chemical exposure can denature the proteins. So denaturation disrupts the shape, conserves the primary sequence, but it conserves the primary sequence. It can sometimes be reversed if the agent is removed, but other times it could be irreversible. Chaperone proteins assist the property of protein folding. They protect and surround the unfolded protein so that all parts are close enough for the interactions to occur. Proteins are amino acid macromolecules. They have diverse cellular functions. Protein structure is organized at 4 levels: primary, secondary, tertiary, and quaternary. And protein denaturation from conditions like temperature or pH changes alter the protein shape, causing a loss of the function. So that was our before the last. That's the protein. What's the last macromolecule we're going to do today? What did we say? There was sugars, carbohydrates, and yeah, the nucleic acids. The nucleic acids. So now, when we examine these nucleic acids, nucleic acids, DNA and RNA, carry genetic instruction for the cell's functioning. So DNA and RNA are polynucleotides. These are chains of nucleotide monomers. These are chains of nucleotide monomers. So each nucleotide contains an heterogeneous base, a pentose sugar, and a phosphate group. So there is a deoxyribonucleic acid DNA. Let's look at the deoxyribonucleic acid DNA. DNA is the genetic material in both eukaryotes. These are housed in the organelles, and prokaryotes, these are in the cytoplasm. So a cell's entire set of genes is called its genome. And the DNA regulates cellular activities by controlling gene expression for proteins on the RNAs. So remember, we said that we have to transcribe the DNA in order to get mRNA. Then we translate the mRNA in order to get, we translate the mRNA in order to get We transcribe DNA to get mRNA, and we translate mRNA in order to get. We translate mRNA to get. We translate mRNA to get proteins in the ribosome. We're going to do that when we do the cell. So DNA regulates cellular activities by controlling the gene expression for proteins and RNAs. Now, when you compare the DNA to the RNA, the ribonucleic acid is involved in protein synthesis using messenger RNA that is transcribed from the DNA. So other RNA types, rRNA, tRNA, microRNA, regulate the protein synthesis. So you see, that is the second carbon, that's the third carbon. On the second carbon, that's how you're going to be able to tell whether or not we have a deoxyribose or oxyribose. And you know the nitrogenous bases come in two families. There is a purines and pyrimidines. Purines have two rings, while pyrimidines have only one ring. So that's the nitrogenous base. So in order to have a nucleotide, you need to have three things. You need to have a nitrogenous base. It could be one of these two. It could be a purine, or it could be a pyrimidine. So that's one out of three. What's two out of three? This pentose sugar. This pentose sugar. This pentose sugar, 1, 2, 3, 4, 5. This pentose sugar is going to be the second component of my nucleotide. And lastly, we're going to have a phosphate group. And that phosphate group is going to, that phosphate group is going to be my last component of the nucleotide. You can never have a nucleotide unless you have nitrogenous base, unless you have a pentose sugar, and unless you have a phosphate group. That is a nucleotide. DNA and RNAs. The DNA nucleotide bases are adenine, guanine, cytosine, and thymine. They are replaced by uracil in the RNA. Adenine and guanine are purines. They have two rings, and cytosine, thymine, and uracil are pyrimidines. So the DNA contains deoxyribose sugar, while the RNA contains a ribose sugar. Nucleotides link via a 3 to 5 phosphodiester bond. So now you see the sugars are connected to each other by a glycosidic bond. The amino acids are connected to each other to make proteins by what kind of bond? Nope. Glycosidic bonds connect sugars together. The bonds that connect amino acids together is known as the. Nope, that is for the nucleotide. That is for the nucleotide. When you want to connect nucleotides together, you connect by a phosphodiester bond. What's the name of the bond to connect amino acids together? Peptide bond. Excellent. That's a peptide bond. You need to know these bonds. Very important. So if you look at the DNA, yeah, we did this in the previous illustration when we were doing proteins. How do we get proteins anabolism if we don't connect amino acids? And we got to connect amino acids through. We connect amino acids through. Yes. Adenine is a double helix with a sugar phosphate backbone, exterior and paired bases. Strands are anti-parallel, running in opposite 5' to 3' direction. RNA is a single-stranded polynucleotide made of ribonucleotides. These are ribonucleic acids. The bases are adenine, guanine, cytosine, and uracil. That replaces thymine. mRNA is transcribed as complementary copy of DNA. mRNA carries the DNA's instructions to ribosomes for protein synthesis. And ribosomes translate mRNA into proteins. They translate RNA into proteins. So how do we get RNA? With base triplets. So we need to have 3 nucleotides that will encode for me which amino acids in the ribosome. So the rRNA, that's the ribosomal RNA. That is the functional part of and a structural and a functional part of the ribosome organelle that's going to enable me to synthesize proteins. It aligns the mRNA and catalyzes peptide bond formation through the peptidyltransferase. tRNAs carry amino acids to the ribosomes. You have anticodons or complementary to the mRNA codons, and microRNA regulates the genetic expression by binding and blocking certain mRNAs from translation. This is the function of the this is a comparison of the function between RNA and DNA. So DNA carries genetic information and RNA is involved in protein synthesis. RNA is involved in protein synthesis. The location of the DNA is in the nucleus, while the location in the RNA is outside the nucleus in the cytoplasm. The DNA is a double helix, and the RNA is usually single-stranded. The DNA has a sugar that is deoxyribose, and the RNA has a ribose sugar. Pyrimidines are cytosine and thymine, and examples of RNA nucleotides that are nitrogenous bases that are pyrimidines. These are your cytosine and the uracil. So you have to understand that RNA does not have thymine. That's an important point. RNA does not have thymine. RNA only has uracil, only has uracil. So nucleic acids, DNA and RNA are polymers of nucleotides, sugar, nitrogenous bases, and phosphate groups. DNA is double-stranded with a helical structure carrying a cell's genetic blueprint, and the RNA is a single-stranded macromolecule and is involved in protein synthesis and regulation. mRNA carries genetic instructions for protein synthesis, and the rRNA, tRNA, and microRNA plays roles in the protein synthesis and regulation. So this is going to be the last part of